In this video, I want to talk about some basic brain anatomy. You've almost certainly seen a brain before. Probably not in person, but maybe in a textbook or a class, on a news article, or even in a horror movie. Let's take a closer look at what an actual brain looks like. Now, this is a preserved brain. It's a black and white photograph, so we're not seeing the color. If we could see the color, it would appear a pinkish beige color. Because this brain's been removed from the skull, I want to just orient you and let you know where some landmarks are. This would be the anterior, the front part of the brain. We also call this the rostral pole. Here's the posterior portion of the brain. We call this the top or superior surface. And down here is the bottom or inferior surface. Now it should be obvious that the surface of the brain isn't uniform. There are regions where there are sort of these bulges and grooves. For example, up here there are relatively large bulges and grooves. Here's a different part of the brain down here, and again, there are folds in the surface of the brain, but you can see that they're much more tight and they seem to be parallel to one another. So we're not the first people to notice that the brain doesn't look the same everywhere, and that led early neuroanatomists to give these different parts of the brain different names. So for example, this portion of the brain here, this large part, is called the cerebrum. Back here is the cerebellum. And here is a region called the brainstem, which actually has several components, and here we can see the pons and the medulla. The images that I've been showing you are from a human, the cerebrum, the cerebellum, and the brainstem, but if you look at these pictures of other mammals, you'll see that we can recognize the same regions. Here's a chimpanzee cerebrum, rhesus monkey, cat, mouse, and over here are the cerebella and the brainstem. Although we can find the same regions in different types of animals, they don't look the same in every animal. Compare, for example, this human brain with this mouse brain. The absolute size of the brains would be very different, but what I want to focus on here is the relative size of the different components. In the human, the cerebrum is really quite large. It makes up about 3/4 of the brain volume, and it's highly folded. In the mouse, the cerebrum's smooth. It makes up only about 30% of the brain by volume. In contrast, the cerebellum is about equally sized in both of these animals, making up about 10%. Because neural tissue is expensive and because we have to fit the brain inside the skull, animals tend to devote more brain space to regions that are functionally important. Here's an example. This large region that I've colored blue at the front of the mouse brain, this is the olfactory bulb, and this is the part of the brain that will be involved in processing odors. It's quite large in the mouse. We can see it in the cat, in the rhesus monkey, in the chimpanzee. In the human, it's present, but it's been overgrown by the cerebrum and it's hard to see. Because different parts of the brain appeared differently, neuroanatomists wanted to know if these different parts served different functions. This was addressed by a number of early neurologists, including Jean-Pierre Flourens. He did something called lesion experiments. A lesion is the term that we give when there's damage to a tissue, and this can occur as a result of an injury or of a disease, but in Flourens' case, he was using it as a tool. So he took animals, mostly pigeons, but also dogs and cats and other animals, and he would look at their behavior, and then he would go and selectively damage part of the brain, and then study the behavior again to see if there were any deficits. And based on these sorts of experiments, he concluded that the cerebellum was necessary for motor coordination and equilibrium. He also concluded that the medulla was essential to life because destruction of the brainstem would lead to death. Well, what about that large portion, the cerebrum? What functions did Flourens ascribe to that? I'm going to go and read to you some of his work. Actually, I'm going to read you a translation of his work because he wrote in French. He said, "If the cerebral lobes are removed, vision is lost, for the animal no longer sees. Volition is lost, for it no longer wishes to move. Memory, for it no longer remembers. Judgment, for it no longer judges. It strikes itself 20 times against the same object without learning to avoid it. It stamps on the ground when struck blows rather than fleeing. Of movements once started continues, but it never begins spontaneously. It only flies if thrown into the air. It only walks if it's pushed. It only swallows if food is forced into its beak. But remarkable to relate, flight, walking, or swallowing once begun continue and take place with perfect regularity. Animals deprived of their cerebral lobes no longer have sensation, judgment, memory, or volition. The cerebral lobes, therefore, are the exclusive site of all sensation, of all the intellectual faculties. What Flourens would conclude would be that the cerebrum, this large part of the brain, is associated with higher cognitive functions, sensory perception, memory, thoughts, judgment, and initiation of voluntary movement. And while all parts of the brain are important, as we continue in neuroscience, we'll see a lot of emphasis on the cerebrum because these qualities are central to know and to be human.